It's a privilege and an honor to be here, so thank you for the invitation. That was a very motivating story, the last speaker, so thank you. My topic is could be viewed as a little controversial, and as much as I like it to go away, it's not going away, and it's only been reified by the current political climate. My topic is based on my personal experiences as a physician, and now as a close relative of mine passed away. One thing I was not mentioned, I was an advisor to President Obama on the precision medicine team, and I'm leading the largest whole genome sequencing study of asthma in the United States. We're whole genome sequencing about 20,000 minority children. And I also teach. I teach pharmacogenetics to all the first-year PharmD students at UCSF, and I run a program in which we teach MDs and PharmDs, postdocs, precision medicine. So without further ado, let me begin. So there's huge debate in medicine and science whether or not we as physicians or providers or researchers should consider race, ethnicity in disease and drug response. And I'll argue both sides of the debate. Many people have argued that race is a social construct. Others have argued that race is a biologic construct, including David Duke, who has contacted me personally. And others feel that race is just a social construct and that has nothing to do with biology, and that is borne out with Michael Brown in Ferguson, Missouri. His death had nothing to do with biology. But at UCSF, we believe that there's an interaction of social factors as well as environmental factors. And I'm a basic scientist, so I'm going to walk you through our beliefs of the biology and the importance of looking at racial differences between populations. Well, these are the demographics of the United States currently, and they're rapidly changing, and as of today, more than 50% of children today that are born are of non-European descent. And the NIH recognized this in 1993, and Congress passed a law called the NIH Revitalization Act, requiring the inclusion of women and minorities in all NIH-funded clinical and biomedical research. And since the NIH does analytics on me as a scientist, we did analytics on them as a citizen. And this is what we published, my colleagues and I, Carlos Bustamante at Stanford and Francisco de la Vega. We published this in Nature a couple of years ago. We analyzed all modern existing data of genetics studies done up until 2009, and we demonstrated that out of all modern genetic studies, more than 96% of the studies worldwide have been done in populations of European origin. That means that less than 4% of the world population was being included in clinical and biomedical genetic research. Well, unfortunately, our colleagues in Seattle did a repeat analysis and published this in Nature in 2016 and demonstrated that that number has increased from 4% to 19%, but what's important to know is that the majority of studies are done in populations of Asian countries, and so the NIH contribution of minority populations went from less than 4% to 6% in the last 7 years. And to me, that's a problem. Problem when you have diseases like common diseases, unlike the last disease the speaker spoke about, this is a common disease. Asthma is the most common chronic disorder of children in the United States. And it also has the highest racial disparity of all chronic diseases. You can see that the populations of the highest prevalence are Puerto Ricans, the populations of the lowest prevalence are Mexicans, and mortality rates parallel that. Yet, we published a paper demonstrating last year that 95% of the NIH budget on lung disease focused in on white populations. And again, so the advances of genetics and medicine are not really reaching the majority US population. So what we asked the question is this genetics or is it environmental? And to that end, we built the largest study of minority children in the United States. We built a large network. We recruited over 11,000 children from all parts of the United States, Puerto Rico and Mexico City, and we wanted to capitalize on the diversity with respect to air pollution, socioeconomic status, as well as genetic diversity. And I'm going to walk you through this. We collected data on genomics, clinical background, socioeconomic status, discrimination, what we call the built environment, as well as detailed measures of air pollution on every single child that we recruited, from the time of birth to the time of enrollment. So we recruited children from 8 to 21. So if you were 8 at the time of recruitment, you had eight measures of geocoded measures of air pollution. If you're 21, you had 21 measures of air pollution measures. So one of the things we did was this. This is called spirometry. And this is really cool. And I'll walk you through this. Here you have a child who has asthma, and we had to get detailed measures, quantitative measures of lung function. And so here we have exhalation on the top, inspiration on the bottom, and we gave them a drug. Here's the first measurement, and here's the second measurement after the drug. The difference in these two measurements are quantitative measure of drug response. And we wanted to use the most commonly used asthma medication in the world, which is albuterol. Whether you're here in downtown South San Francisco, New York, Tokyo, or Dubai, if you have asthma, you're going to get albuterol. It's the most commonly used drug, particularly amongst poor children. And this is what we found. This is cool. And we published this. We were one of the first to demonstrate that there are racial differences and ethnic differences with respect to drug response. I'll walk you through this. On the y-axis, we have drug response. The higher you are, the better. On this axis, this dotted red line is where we as clinicians say you've had a therapeutically good response. And what we found is that the strongest predictor of drug response for these children was ethnic background. So the population with the highest prevalence and mortality also had the lowest drug response to commonly used asthma medications. Well, we thought we were one of the first to publish this. But lo and behold, GlaxoSmithKline was on the hunt. And they published their data, but they published their data in this. This is the package insert. It's probably the first thing you throw away when you get your medications. I don't know, ask yourself, how many of you have ever read one of these? Well, this is the package insert for one of the major asthma medications called Advair. And I'll tell you why you don't read them. Because this is what it shows. And I've highlighted in yellow and have a large red arrow. But if you read closely, this is what the package insert says. If the African-American asthma-related deaths occurred at a rate 7 times that of whites. So this medication is still in operation, but they modified it, so now you have to take an inhaled corticosteroid to offset the positive effects or the negative effects of a positive medication. So now we have to try to understand population genetics. And this is where my talk will take a particular twist. As you know that we're all descendants of Africa. There's migrations around the world. And these migrations go back to 200,000 years ago. But since we were studying contemporary populations like Hispanics or African Americans, and it could be Filipinos, it could be Asians, it could be Indians, but I'm going to focus in on Latinos and African Americans and just use them as an example here. Latinos and African Americans really are a young population. They consist of the number 1 and 2 largest minority groups in the United States, approximately 40% of the US population. They really were created in 1492 when Christopher Columbus came to the New World and led to the massive, largest importation of African slaves to the New World. So the contemporary US population, or worldwide, is an amalgamation of 3 ancestral groups: African, Native American, and European. And the contemporary group, what we call racially admixed. And I created this slide in 2000 when we started this work. And it's basically to show that each individual, even though they may self-identify as one particular group, say Hispanics, they actually vary from percent to one individual to another individual. So we use the techniques that long before 23andMe started, long before Ancestry.com started. This is going back to 2001. And what we did is we measured genetic ancestry. And I know each of you intuitively knows what this is because you've all watched Crime Scene Investigation or CSI, kind of cool. There might be blood on a crime scene, a forensic pathologist comes in, isolates DNA, measures the DNA for genetic markers, and aligns it to genetic samples collected from around the world. So for example, if I had my DNA measured, and I did through 23andMe, I'm 26% Native American, 6% African, and the rest is European. When I'm going back to this particular study, where we had 11,000 individuals, and we took a subset of individuals, and we measured genetic ancestry in 3 groups, African Americans, Mexicans, and Puerto Ricans. Each bar represents an individual. Each color represents that individual's ancestral component. So green being African, yellow being European, purple being Native American. And you can see in this individual here, and these individuals all self-identified as being 100% pure Mexican, African American, or Puerto Rican, and all 4 of their grandparents. So as you go from left to right, I'm Mexican. If you go from left to right, as I mentioned, I'm 26% Native American, 8% African, and the rest is European, so I'm an undercover brother. So, but this is interesting and kind of recreational. But here's where it gets really interesting, and I want to use a fascinating story. This is an African American, and these are your karyotypes. And this is, in the United States, average African Americans are 80% African, and the rest is European. Well, there's a beautiful example related to multiple sclerosis that occurred at UCSF. Multiple sclerosis is a neurologic disorder that primarily occurs almost exclusively in populations of European origin. But we know African Americans do get multiple sclerosis, like the talk show host Montel Williams. So Steve Hauser and Jorge Oxenberg paired up with Montel Williams and hypothesized that MS, or multiple sclerosis in African Americans, was due to the European gene coming through the population. So they, in collaboration with Montel Williams, they created the largest study of African Americans with multiple sclerosis. They assigned genetic ancestry at every area of the genome, just like what 23andMe does. And here's what they found. They got a home run. They found that every African American that had MS also inherited a European chunk on chromosome 1. And now with the advent of whole genome sequencing and targeted sequencing, they're fine mapping this region. So there's a lot of work that's going to be coming down the road. Well, we've done the same thing in all of our Hispanics and African Americans. We just published a nice paper in Nature showing that women who had breast cancer, who are of Hispanic origin, who inherited a European or a Native American chunk at the estrogen receptor, were protected from the development of breast cancer. We've done that for many phenotypes related to asthma. So on chromosome 6, if you've inherited Native American ancestry, you had lower levels of IgE, which is a biomarker of asthma. And I'm going to show you that on chromosome 10, in one of my next examples, that Asian ancestry at that particular region of the chromosome at a gene called CYP2C19 confers resistance to one of the most commonly used drugs in the world for heart attacks and strokes. So this is where it takes a personal twist. This is Plavix. This is the #1 drug for heart attacks and strokes. And if any of you have a heart attack or a stroke right now, and you go to any of the major hospitals in the local area or nationwide, you're going to get it whether you like it or not. And in part, it's because Bristol-Myers Squibb and Sanofi Aventis were able to get it at a good deal on all the Medicare formularies and all the hospital formularies in the United States and around the world. So the question is, are they gambling with your life? And Plavix is an interesting drug. It's a prodrug, and it needs to be activated in order for it to work by genetic polymorphism. Well, that genetic polymorphism in a gene called CYP2C19, cytochrome P450-219 on chromosome 10, is absent in 50% of Asians, 75% of Pacific Islanders. Despite that, the company got it marketed on formulary in Hawaii, which, as I'll remind you, is a Pacific island and also has a lot of Asians. So the Attorney General, David Louis, a Chinese lawyer from Oakland, California, sued Bristol-Myers Squibb on behalf of negligence. They had the data demonstrating that the gene or the allele was absent in most Asians and Pacific Islanders. Despite that, they went ahead and got it on formulary. And these are reports from 2014, and Bristol-Myers Squibb has a staff of lawyers that are probably in the hundreds of thousands. The Attorney General's Office of Hawaii has a staff of lawyers in the number of 12. As an appointment, the Attorney General is an appointed position was reassigned. So that case is mired, and in the courts, it's a David versus Goliath example. And I have a very interesting background. Although I'm Hispanic, I live with a Chinese family, went to Chinese school. These are what I consider my brothers. I grew up with them. I went to Chinese school in Chinatown for 2 years. This is Marco, who's pretty healthy. Recently passed away on August 24th of last year as a result of an adverse drug reaction. So even though I teach pharmacogenetics to hundreds of students, and we just had my class last Tuesday in which we provided genetic test results to all of our students based upon their ethnicity, and we gave them counseling as to which drugs they would work against, which drugs would work for them and which drugs wouldn't work for them. Within my own personal family space, Mark died of an adverse drug reaction to a cardiovascular medication less than a year ago. So this has taken a personal twist to me and now has galvanized a lot of the work that I do. So it's not just academic or theoretical, it's now personal. And basically what we're trying to get to is we believe that there's one-size approach does not fit everybody, and that we need to have targeted therapy. As you heard with the last presentation, targeted whole genome therapy identified a new diagnosis. There are different flavors of disease, or what we call clinical endotypes, that could be stratified by sequencing or genotyping. And with that, I'll end there to be on time. But I want to thank you for the opportunity to present, and I'd be happy to take any questions.